the recent advancement in the field of homeopathic pharmacy. Though I'd like to speak not on recent advancement, I used to drop the word recent. It is the advancement in the field of homeopathic pharmacy since the time of Hahnemann. We all know that pharmacy, though the most neglected subject in homeopathic curriculum, has immense importance to establish effectivity of homeopathic medicine to cure the diseases. We all know that. But still it is the most neglected subject in the curriculum. And no treatment will be effective in spite of correct diagnosis and prescription if the administered medicine is bad in quality, isn't it? So start the talk, let us understand what is pharmacy. The pharmacy derives its name from the Greek root pharmakon, a drug. And the pharmacy is concerned with manufacture of the drug, its formulation, quality control, and dispensing of medicaments to treat the disease. It is a very, very vast subject. So I shall divide my discussion into 4 parts. The first is identification of raw material and its quality. Second is advancement in the field of manufacture. Third is standardization of homeopathic medicine. And the fourth, understanding potentization. Let's take the first question. Identification of raw material and their quality. We all know that homeopathic medicine is prepared from 7 classes. The first from plant kingdom, from animal kingdom, mineral kingdom, synthetic chemical compounds, nosodes, sarcodes, and also on impongal area. Today I will stick only to plant kingdom. I told you that it is a very big subject, very vast subject. So today I will stick only to plant kingdom. Identification of raw material obtained from plant kingdom. How to identify the material. Till today, 6 major points for testing suitability of plant material is taken as identification of general up to species level, finding morphological and organoleptic variation of the drug, finding purity and freedom from adulteration, finding methods of cultivation, finding suitable method of collection and storage, and study of constituent and the standardization. Here we all know that. You shall have to identify when we try to identify a drug substance, you shall have to identify it to species level. That means when you are identifying belladonna, say belladonna, the only one belladonna is not enough, it should be called Atropa belladonna. Atropa is the genus and belladonna is the species. But how to identify that? Usually it was being identified by morphological and organoleptic variation. There are so many morphological organoleptic variation, and we are still identifying it through morphological variation. But so there are other methods of identification also. We'll go to that. Secondly, the material what we have been collected came to us. It should be pure and free from adulterant. So we must have knowledge about the adulteration that is given in the drug. What I've said, that finding method of cultivation. You may question why the method of cultivation is important here. Yes, we will also come to this question later on. Finding suitable method of collection and storage, that is also important. We'll all come to these points later on. So let me first concentrate on. No, before that, I'll say that though these were being tested till date, 4 new parameters are coming through. As I am a member of Pharmacopoeia Committee and Standardization Committee of CCRA, I'm telling you that 4 new parameters are coming to identify a drug. That is testing for heavy metal, testing for microbial contamination, testing for presence of aflatoxin, and testing for presence of insecticides and pesticides. The drug, botanical material, what we are getting nowadays, we find that they are contaminated with heavy metals, especially if we collect some drug from side of the roadside. They are heavily contaminated with lead, and that lead, if comes into our medicine, that will be dangerous. Microbial contamination, if you can't store it properly, store the drug properly, microbial contamination will adhere. Test of aflatoxin, if it is fungus is, sometimes we find that fungus are there on the drug and aflatoxin are being produced. And for microbial contamination of aflatoxin, so many drug suppliers that putting insecticides and pesticides on the drug, that is very dangerous. So these 4 parameters are also be tested so that we can get good drug, good quality drug for making of our medicine. Till date, the pharmacoprognostic evaluation of drug, we are depending on 4 factors. There is organoleptic studies. I told that organoleptic studies, that means macroscopic studies of the drug material. Then microscopic study, that is section study and powder microscopy. Third is physical analysis. That means acid value of the drug, ash value of the drug, saponification value of the drug, and chemical analysis. That is phytochemical examination, chromatographic, TLC is a thin layer chromatography, it will be GLC, gas liquid chromatography, or HPLC, high performance liquid chromatography, or even on spectrophotometric studies as UV or IR. Even ask me one question. When we can examine the drug chemically, whether the drug is right or wrong, why the organoleptic study and microscopic studies and all those studies are required? It is required as because morphological studies, raw drug material of plant material origin have to be put under morphological and microscopic studies. Chemical studies alone is not sufficient, as same active principle may be present in different species, but those different species may show different biological activity. You see, take for example, Barbaris vulgaris. It contains berberine. You know it contains berberine. If we test some drug and if we find it contains berberine, then we'll be sure that this Barbaris vulgaris. No, it cannot be. It may be Barbaris accifolia. Even it may be hydroxys even. Completely different drug, but they also contain berberine. So only on content of chemical content of some drug, till date we cannot identify a botanical drug. So we shall have to go for organoleptic and microscopical studies. Organoleptic study, morphological organoleptic study is open, very easy to naked eye. We are studying it naked eye. We are thinking only if it is leaf, the shape of the leaf and all those things, you know it very well. But what about the microscopic studies? Till date, what we are doing, we are putting the slide under the microscope, putting our, and there's a picture of this is a microscope. We are putting the slide under the microscope and when examining through these eyepieces, putting your eye on the eyepieces, see what is the cell structure and comparing it with oak. That is a very difficult thing to do. Nowadays, what is being done nowadays?
Sir, sorry to interrupt you.
Yes,
your slide is not moving.
Is it so?
Slide. Yes.
And
you can do the full screen now.
Oh yes.
On full screen, yeah.
No, it was on full screen. Is it moving now? Is it moving now? Yes, sir.
Hello. Yes, sir.
Is it moving now?
Yeah.
Okay.
Yes, sir.
The microphone.
Okay. Till date, what we are doing is we are putting our eye in the microscope eyepieces, looking at the cell structure and then comparing it with the book picture of the book. But nowadays, what we are doing, we are putting a camera on the microscope. The camera brings the picture in the computer, and if we have standard picture in our computer, we can compare it very easily. Here I am showing Echinacea angustifolia stem transverse section. This is the authentic sample picture we have in the computer, and what is given in the under the microscope. It is here. The picture is here. So it is very easily we can compare these two very easily. I told that knowledge about common adulterant is also important, as because what we are seeing, we find that very common drugs are being adulterated. For example, Passiflora incarnata. No, it is not a very common drug, of course. It is a totally imported drug. But here in India, we can get Passiflora indica, a different species, and it is being adulterated very often with Passiflora incarnata. We must be very sure what is the difference of cell structure of Passiflora incarnata and Passiflora indica. Raoulsia serpentina is an Indian drug, very Indian drug, but still, what we find that in the market, we find at least 50% of Raoulsia serpentina is not Raoulsia serpentina, it is Raoulsia cannabis, more easily grown Raoulsia species that doesn't contain Raoulsin and all other chemical ingredients of Raoulsia serpentina. Ashoka bark. The Ayurvedic people are using tons of Ashoka. But do you know, at least 80% of them are not Ashoka, they are Polyalthia longifolia. Ashoka is not very abundantly available in India. Ashoka is scarce. Another is of animal origin, Lita vesicatoria. Cantharis, they are very well-known cantharis. This cantharis is available no more. And what people are using cantharis is Mylatis, some Mylatis species. So we must be very sure the drug what we are using, whether it is adulterated drug or has been replaced by some other drug. So leaving about the identification part, let us come to the advancement in the field of manufacture. The time is very short, so I shall have to go very fast through all those things. For the advancement in manufacturing, I am only talking about mother tincture, no other thing other that will be taken afterwards. The new method has been adapted for preparation of mother tinctures. But how that differs from Hahnemannian method of preparation of mother tincture? Do you know? In the Hahnemannian method of preparation, juice of fresh plant was used, or juicy plant, less juicy plant, and dry plant is segregated into four classes.
The slide is not moving. Yes.
Let me keep it in this way. Okay. Otherwise, it is studied.
Yeah.
Is it visible now?
Yeah. Visible but not moving now. When you describe the so just you move the slide also.
Okay, that's okay.
Yeah.
In the Hahnemannian method, the botanical drug substances are classified into 4 different groups. That is juicy plant, juice of the plant, less juicy plant, and dry plant. But during transportation of to a distant place, this classification no longer holds good. Just think about the juicy plant, thuja. It contains, at least in juicy plant, it contains something like 75 to 80% of plant moisture. You have collected some thuja somewhere. If you prepare medicine from that plant, juicy plant, according to Hahnemannian method, you will get a mother tincture. You transport that thuja to some 100 kilometers away, it will get dried. And there, if you prepare mother tincture of that thuja, the drug concentration in the mother tincture will definitely differ. The problem is a standard of produced mother tincture will differ. There is a problem with the Hahnemannian method of preparation. So now it has been coming, we are coming to new method of preparation where solid content of a plant material is taken as the unit of medicinal strength. Not the whole plant, we're taking the moisture content of the plant is not a drug substance. Only the solid content of the plant material is a drug substance. And volume of the mother tincture is calculated on that very unit of solid content. a standard of produced mother tincture will never differ. If you take the solid content of thuja, if it contains 80% moisture, the solid content will be 20%. That solid content will never differ if you transport it from one place to other. So standard of mother tincture will not differ. So that's why almost throughout the world, everybody is now using this new method of preparation of mother tincture. But here I may ask you one question. You have seen that for people in the mother tincture, so many percentage of alcohol are being used. Do you know why? Why alcohol? Why alcoholics in the mother tincture differ from drug to drug? And how to ascertain at which alcoholic concentration mother tincture a new drug is prepared? You must say very easily, yes, it is given in the pharmacopoeia, so we will use that concentration of alcohol. But if you have to prepare a new drug, new mother tincture, how is it going to ascertain?
अगर तो कुछ स्टेटमेंट भी पढ़ रहे थे हमारे फोन मार
Sir, will you please keep the mic closed?
अब आता है
Yes, sir. Stand visible.
Okay, it's okay. Okay. I'm sorry for the disturbance. There is something wrong with the computer. Hi, sir.
No, sir. It's okay, sir. Continue, please.
It's okay. What we are talking about, why the new method is being adapted, and I finished that. I think I finished that. Okay. Calculation of margin that has also been put. Maceration and percolation. Yes. We will stop here. Which method is to be followed for preparation of mother tincture? Is it maceration or it is percolation? Percolation is without a faster process, but not suitable for all drug substances. You must be sure, and you must first of all you must test with that whether that drug material is suitable for percolation. Otherwise, maceration is universal process. We will go for maceration process.